Thank you for joining us today for the Innovators in Cognitive Neuroscience speaker series. Before we begin, let us please acknowledge the indigenous lands from which we are calling in today. The land I am calling in from and the surrounding area is marked in name by the people from which this land was taken, the Massachusetts people. Please refer to the link in the chat to take a moment to reflect on where you're joining us from today. Through the recognition of past harms, we can resolve to create more equitable and humane futures. Hi, everyone. My name is Daniel Giannini, and I'm a graduate student at Harvard University. On behalf of all the students and postdocs of the organizing committee, I'm happy to welcome you to the 6th talk of the Innovators in Cognitive Neuroscience speaker series. Collectively, we represent a consortium of schools committed to highlighting innovative advances in cognitive neuroscience, including Columbia, Dartmouth, Gallaudet, Harvard, MIT, UPenn, Princeton, and Yale. We acknowledge that these schools are elite institutes of higher learning, and in order to de-silo the generation of knowledge within the ivory tower, we plan to make these talks publicly available on the website with the consent of the speakers. Which brings me to today's talk. Today we'll be hearing from Dr. Alexandra Keinath. Alexandra studies the role of the hippocampus in spatial navigation and memory. She completed her undergrad at Carnegie Mellon, followed by her PhD in behavioral neuroscience at University of Pennsylvania, where she worked with Russell Epstein. Alexandra is currently a Banting postdoctoral fellow at McGill, working with Dr. Mark Brandon. Her research includes varied methods of neural recording, circuit manipulations, and computational approaches, and her work spans both mice and humans. She's studied a wide variety of topics, including the neural mechanisms of spatial reorientation, the representations of ventral hippocampus, and the grid-like coding of visual space in human entorhinal cortex. Today, she will be presenting her work titled Experience-Dependent Dynamics of Hippocampal and Entorhinal Spatial Representations. In addition, she will be giving her talk in support of the Mazzoni Center. Please join me in welcoming Dr. Alexandra Keinath.
Hi, everyone. Perfect. Thank you so much for that introduction. Should I start by sharing my screen? Okay. Let's start there, and then let me just take care of some things. All right. So hello, everyone. I'm just going to assume that y'all can hear me. As you could see, at the midnight hour, I kind of changed up the title of my talk. So if you saw the previous title, sorry. I promised all the work that I said I talked about in the abstract will be presented here today. I just had some additional room, so I wanted to talk about some more topics as well. But before I begin, I'd like to take some time to say a few words about access to healthcare for trans people. To be honest, I've never really done something like this before, and I feel a little unqualified, but I think the visibility is important. And having been given the unique opportunity to talk about an issue like this in front of a scientific audience, I felt the need to take it. Trans rights and access to healthcare are both fundamental human rights. In the case of trans people, this includes access to gender-affirming care. Now, though our community is generally underserved in numerous ways, one avenue of support is clinics such as the Mazzoni Center in Philadelphia, which happens to be where I get most of my care. The Mazzoni Center provides access to gender-affirming healthcare with support for the uninsured, medical care more generally, as well as counseling and social support programs for trans folk of all ages, including youth and their families, as well as programs specifically geared towards trans women of color. The center organizes an annual trans wellness conference to facilitate information exchange and access among providers. And most of the support comes from members of our own community, with almost 30% of the Mazzoni staff identifying as trans or non-conforming. And although I'm highlighting the Mazzoni Center for all the good it's been doing, I'm sure there are other such clinics scattered about. So please keep in mind the needs of this community and consider supporting those who support it with your time and talents if possible. It literally saves lives. Also, before I begin, I'd like to thank the institutions where this work took place, the fellowships which have and are supporting me, the funding for the labs more generally, and the people with whom I've worked on these projects, including Russell Epstein and Vijay Balasubramanian, some of my advisors from my PhD, and Mark Brandon, my postdoc advisor. I'd also love to thank the organizers of the series for having me. I'm super honored and humbled to be here. And if my mom remembered all the Zoom practice, I'm pretty sure you're making her day right now too. Okay, let's talk about some science. So hippocampus, entorhinal cortex, I don't know, you might have heard of them. What spatial representations are we talking about here? Well, decades of experiments have revealed that the hippocampus represents space through the coordinated activity of place cells. In most of these experiments, neural activity is recorded from an individual place cell as a rodent explores an environment. Typically, the activity of this place cell is highest when the animal is in one location of the environment, represented here as a warm color at the analogous pixel location of the rate map. When the animal is in a dispreferred location, activity will be low or zero, represented here as a cool color at the analogous pixel location of the rate map. In this way, a rate map will summarize the time average spatial activity of a single cell. Now, different place cells prefer different preferred locations, spanning the navigable space, collectively forming a quote-unquote hippocampal map of the current environment. Now, one of the reasons why we get so jazzed up about the hippocampus is that each hippocampal map is somewhat unique to the navigational context. For example, if you record the same cells here in a similarly sized square room in a square environment in a new room, you'll see changes to the hippocampal map, with some cells firing in new preferred locations and other cells turning on or off entirely. These differences are known as remapping. Notably, remapping is not a singular phenomenon, but can take multiple forms. For example, simple sensory changes to cues, such as the color of the walls, often induce less drastic remapping, where some cells exhibit no significant changes to their tuning properties, while other cells will fire at the same preferred location, but do so at different rates. Now, given these differences, some have made categorical distinctions between types of remapping, global versus partial versus rate, and indeed there's some evidence that changes to entorhinal inputs might differentiate more extreme forms of these categories. But there remains a debate. Complicating matters further, more extreme versions of rate remapping can mimic changes in preferred locations, blurring the lines between these categories. Since I have the pulpit, I'll say that my own view is that remapping lies on a continuum, and some of the data I'll present today will speak to this. At the very least, we will all agree that we could quantify the extent of remapping to assay the representational similarity in a variety of conditions throughout the hippocampal circuit. So this gets us up to speed with what we need to know about the hippocampus for our purposes today. What about spatial representations in the entorhinal cortex? Well, the entorhinal cortex, and in particular the medial entorhinal cortex, is full of fascinating spatial cell types, perhaps the most famous of which is grid cells. Grid cells fire at a hexagonal lattice of locations tiling the entire navigable space. Each grid cell can be described with a few parameters, such as the orientation of its grid, the spatial phase of its grid relative to environmental boundaries, and its scale, that is, the distance separating neighboring grid vertices. Moreover, the grid network is modular, with different modules of grid cells having different spatial scales. These modules are also anatomically organized such that small-scale grids are observed more dorsally, while large-scale grids are observed more ventrally. Now, grid cells aren't the only spatial cell type in the medial entorhinal cortex. In addition to grid cells, there are also border cells, aka boundary vector cells. These cells tend to fire when a navigational boundary is nearby and in a particular allocentric direction. Thus, they can track changes in environmental geometry when a boundary is elongated or inserted. Together, the coordinated activity of these and other cell types are thought to instantiate some sort of cognitive map. Now, there's a lot of debate about how best to describe this map. Is it Euclidean? Is it simply graph-like? Does it map transitions? Does it depend on scale? Is it a successor representation? I'm not going to get into all of that today. What I will say is that there's something very map-ish about these representations, especially in the small-scale spaces that we've been able to explore in rodents. And I think that this map-ishness has really done a lot to serve our intuitions about what's going on in these regions. Nevertheless, there are some problems with the map metaphor. One problem that I'd like to highlight today is that the idea of a map is typically that of a static representation of the world. Yet, our experience of the world is not static. It's intrinsically dynamic. Sometimes the world changes, and sometimes our experience of it changes. And if our spatial representations want to do a good job of keeping us alive, then they need to be able to adapt, incorporate, and represent these dynamics. So today, I'm going to tell you about some of my work from my PhD and postdoc addressing the question, how do hippocampal and entorhinal representations adapt to a dynamic world? In part one, I'm going to show you that changes to the shape of an environment induce dynamic experience-dependent shifts in rodent grid cell representations and human memory alike. In part two, I'm going to show you that BGCA3 circuitry within the hippocampus mediates a representation of recent experience, even in the absence of disambiguating cues and explicit task demands. In part three, I'm going to show you that hippocampal pattern separation and completion dynamics richly develop over more than a month of protracted experience. So let's turn to part one, grid cells. Now, numerous pieces of theoretical and empirical evidence have linked the grid cell representation to path integration. That is, the use of self-motion information to update a representation of a navigator's current location. One such piece of evidence is the observation that a relatively simple attractor network model performing path integration is capable of recapitulating a grid-like representation. Now, without going into too much detail about in this model, a grid-like pattern of activity is translated on a neural sheet in concert with the movement of a navigator. A gain factor controls the amount of translation with the speed of the navigator, allowing one to model multiple modules of different spatial scales. Oh, my computer is having a real hard time with this GIF. So it will speed up. So now this solution of performing path integration works really well for some time, but there's a well-known problem with path integration. That is, any errors in updating will continue to grow and grow with time unless the path integrator can be reset by an external cue. One suggestion has been that environmental boundaries might be such a cue. By this logic, as the navigator explores the environment, path integration error in the grid cell network will continue to grow and grow until a boundary is encountered. Once encountered, the spatial phase of the grid representation is reset and path integration can proceed from there. So this sort of solution works really well as long as the environment remains completely static. But what if the environment changes? Well, in that case, we and others noted that this boundary-based error correction mechanism makes some pretty unique predictions. Let's unpack them here. Here I'm showing you a grid cell in a standard familiar square environment. I've marked the spatial phase of the cell to the west boundary in red and to the east boundary in blue. In the familiar environment, these two phases are commensurate. Now let's consider a case when the environment is compressed and our boundary-based mechanism continues. Well, by virtue of our boundary-based error correction mechanism, we would expect the spatial phase of the grid cell relative to the west border to be preserved after contacting the west border. Likewise, we would expect the spatial phase of the grid cell relative to the east border to be preserved after contacting the east border. And the combined time-averaged rate map should then be some averaging of these different phases. We can also consider a stretching deformation. Again, by virtue of our boundary-based error correction mechanism, we would expect that the spatial phase of grid cells relative to the west boundary should be preserved after contacting the west border, and the phase relative to the east border to be preserved after contacting the east border. And that path integration just kind of fills things in the newly accessible territory. Thus, in this environment, the combined time average rate map should then be some averaging of these different phases. We can see this borne out in a model when the data are similarly divided by east and west boundary contacts. Crucially, however, all these dynamics would be washed out if we considered only the time averaged rate maps. And indeed, prior to the work I'm presenting here, that's all that was published. So let's check out some of those time averaged rate maps now. At the time of this work, there were two primary data sets investigating the influence of environmental deformations on grid cells. The earliest, Barry et al. From the Jeffrey lab, reported that the grid pattern undergoes some amount of rescaling, matching the deformation of the environment. In the second publication, the Stensola et al. From the Moser lab, they reported a somewhat similar rescaling effect. However, they noted that rescaling was observed only in large-scale grid modules, with small-scale grid modules failing to show evidence of rescaling in their paradigm. I'll note that these results were the primary evidence that individual grid modules were functionally independent. But as I noted before, any interesting boundary-based dynamics would be obscured by time averaging. Moreover, our own modeling and the modeling of other labs indicates that such dynamics could contribute to the appearance of rescaling and could even capture the relationship between grid scale and rescaling without asserting functional dependence. So, show me the money. Are dynamic boundary-dependent shifts contributing to these time-averaged results? To address this question, we reanalyzed data from both data sets. And not that either of these labs really needs a shout out from me, but shout out to them nonetheless for sharing the data. So what do we see? Well, here I'm showing you the time averaged rate maps of a single cell from the Stensola data set in the familiar and compressed environments. It's a large scale grid and shows the appearance of rescaling as they report. Next, we divided these data according to the most recently contacted boundary, contrasting east and west contacts. When we do this, we see that in the familiar environment, spikes following the east and west contacts tend to fall in roughly the same location. But in the compressed environment, we see a shift in these relative spike locations, just as predicted by our model. We could quantify this shift by computing the cross correlation between east and west boundary rate maps. When we do this, we find in the familiar environment, no shift is the best alignment, indicated by this peak in the center of the cross correllogram. But in the compressed environment, the optimal shift is a pretty large shift in this case. Pretty wild. Let's check out another example. Here I'm showing you a small scale grid, which did not show the appearance of rescaling from the same data set simultaneously recorded. Although the boundary condition spikes and rate maps appear a little messy, the cross correlation tells a clear story. During the compression, we see a shift between the east and west boundary rate maps. And I'll show you one more example pretty quickly because they're pretty. Here's an example from the Barry's data set that shows some amount of rescaling. And when we divide the data by north versus south, we see a clear shift in the alignment between these two rate maps. And crucially, we do not see any sort of shift on the northwest axis when only the east-west axis is compressed. Now, of course, we need to quantitatively test our predictions of our model. And while I don't have time to go through them all today, I will say that we tested a bunch of predictions just listed here, all of which were borne out in the data. And you could read about this. It's all out now in eLife. And to close out this section, I want to note that we also noticed that these dynamics, in concert with other work, motivated an additional prediction that spatial memory might also show boundary-dependent shifts during environmental deformations. We sought to test this prediction in follow-up work. To this end, we taught participants the locations of objects in a virtual square environment and later tested their memory in stretched or compressed versions of this environment. And indeed, across 3 different manipulations, a desktop VR version where we forced them to rely on path integration, and an immersive VR version, we observed that predicted shifts in human spatial memory, just as we expected. Pretty exciting. This recently came out in Hippocampus, so if you want more details, feel free to go and check it out. So let's turn to part 2. DGCA3 circuitry mediates hippocampal remapping on the basis of recent experience. Now, as I noted in the introduction, our experience of the world is dynamic. Sometimes the world changes, while sometimes it's our experience of it that changes. In this project, we sought to test whether the hippocampus might differentially represent the same spatial position on the basis of recent experience, even in the absence of disambiguating sensory cues and task demands. To this end, we needed a recording technique and a paradigm. To record in this and the next project, we relied on calcium imaging. Briefly, our flavor of calcium imaging involves the use of miniature head-mounted microscopes to measure the simultaneous activity of large populations of neurons in awake, freely behaving mice. Whereas with standard electrophysiological techniques, we would be lucky to simultaneously record tens of neurons. With calcium imaging, we can literally record hundreds of cells. Moreover, we can do so over weeks, tracking the identity of individual cells as we go. This gives us the power to monitor the population activity with a new level of precision and across previously inaccessible timescales. So, recording technique, check. Next, what paradigm might induce remapping without changes to sensory cues and without explicit task demands? Well, after going through the literature, we stood on the shoulders of giants and decided to rely on the age-old method of putting a mouse in a box. This box. Here, you'll note that there is a central chamber with two different entryways connected by an L-shaped hallway. The mouse is free to run around as he chooses, and sometimes he'll enter the chamber from one entryway and sometimes from the other. Once in the chamber, however, there are no sensory cues disambiguating the points from which the mouse entered. Now, based on prior work examining how entryways determine hippocampal representations across environments, see below, we hypothesized that the mouse might differentially represent the same locations within the chamber depending on the point of entry. And to be super clear, there's no explicit task here. The mice are just freely and happily running around. They're not foraging for anything. They're not doing any specific task. So what do we see in CA1? Well, here I'm showing you 8 simultaneously recorded example cells when we divide their data according to the most recent entryway and session half. You could see that each cell tends to fire at the same location regardless of entryway, but appears to do so at different rates. But these are just some examples, and we could quantify this. But before we do, we want to be extra sure that any differences we measure are not driven by sampling biases associated with the point of entry. So to control for this, what we're going to do is randomly subsample our data to match the spatial sampling distributions across all comparisons. And with this in mind, we first computed the population vector correlations of rate maps across both halves of the section when the mouse entered either from the same entryway or different entryways. When we do this, we find that the hippocampal maps are more similar over time when the mouse entered from the same entryway rather than a different entryway. Moreover, when we look at the firing rates alone, we see a similar story. So indeed, it looks like the hippocampus differentially represents the same location on the basis of recent experience in the form of rate remapping. We next wondered what circuitry might mediate this experience-dependent representation. To address this question, we need to fill in a little more information about the hippocampus. So the hippocampus is a beautifully stereotyped circuit. It's my favorite for a reason. It grossly consists of 3 basic elements: the dentate gyrus, CA3, and CA1. Information flows through the circuit in 2 ways, primarily. Firstly, there is a direct pathway from entorhinal cortex, the major input to the hippocampus, to area CA1. Secondly, there's a more circuitous route from entorhinal cortex to dentate gyrus, from dentate gyrus to CA3, and finally from CA3 to CA1. This route is aptly termed the trisynaptic pathway. Now, previous theoretical and computational work has implicated this pathway as a potential source of rate remapping and as a hub where experience might be represented. Thus, we hypothesize that this pathway might mediate the representation of the most recent entryway that we observe in CA1. We first reasoned that if indeed this pathway is the source of entryway remapping in CA1, then a similar representation should be observed just upstream in CA3. To test this, we recorded from CA3, and indeed, we saw a similar, if not slightly stronger, entryway remapping effect. Great. So trisynaptic circuit could feasibly be conveying this information to CA1, but does it? To answer this question, what we really need is a causal test of this hypothesis. And to this end, we turn to a chemogenetic dread approach. Specifically, we express the inhibitory designer receptor HM4DI in the dentate and CA3 of GRIC4 Cree mice via viral Cree-mediated transfection, and we did this expression bilaterally and longitudinally. This receptor allows us to inhibit the trisynaptic input to CA1 through the systemic administration of the drug CNO targeting these receptors. So now we're going to be able to record from CA1 in our paradigm as we turn off or leave on the trisynaptic circuit. And of course, because we're using a drug, we're going to want to start with controls to make sure any results we observe aren't due to off-target effects. So here I'm showing you data from controls without the HM4DI receptor. Now, whether or not we give them saline or CNO, we observe the same entryway remapping effect. Now let's turn to some experimental animals. When we give these experimental animals with HM4DI in their trisynaptic circuit, when we give them saline, they continue to show entryway remapping as before. However, under CNO, this entryway remapping is abolished. We see the same thing when we look at the firing rates alone. And as an aside, I think it's pretty incredible to have a null result with 1,800 cells. It's statistically hard. And I'd also like to note that entryway remapping in this case is not being eliminated by damaging the hippocampal representation. Rather, it's being achieved by an increase in the similarity of different entryway maps. In line with this, when we check other measures of place code quality, such as firing rates, spatial information content, or split half reliability, we see no differences between saline and CNO in our experimental mice. So together, these results indicate that the trisynaptic circuit plays a causal and selective role in mediating remapping in CA1 on the basis of recent experience. And I'll note that there's a bunch of additional results that I don't have time to unpack here, so I'm just listing them. If you're watching on the replay, you could pause here. But this work is out now in NatCom. Feel free to check it out there. The supplement has more figures than my entire PhD thesis, so clearly there's a lot more analysis going on. Okay, now let's turn to part 3, some new and unpublished data. Hippocampal pattern separation and pattern completion across protracted experience. Well, what do we mean by pattern separation and pattern completion? Let's start with pattern separation. Pattern separation is a term used to describe a functional mapping of inputs to outputs, but not just any mapping. Rather, it's a mapping of similar inputs to dissimilar outputs. For example, imagine that I, as I often do, map words to conceptual outputs. In this case, I receive two similar input words, car and cat. Now, when I receive car, I call to mind something like this. But when I receive cat, I call to mind something like this. Thus, I am mapping similar input words to dissimilar conceptual outputs. Now, pattern completion is the complement of pattern separation, where different inputs are mapped to similar or even the same output. For example, when I receive either car or automobile, dissimilar input words, I call to mind the same 1987 black Lincoln Town Car in both cases. Hence, I'm mapping dissimilar inputs to similar conceptual outputs. Now, these processes are thought to be important at a variety of stages of neural processing throughout the brain, but are thought to be especially critical to successful episodic memory, governing phenomena such as recall, memory interference, and generalization. Indeed, decades of research has built entire models of memory around these processes, often tying them to specific anatomical circuits. Suffice it to say that understanding whether and how pattern separation and pattern completion shape neural representations in the hippocampus is of central importance to a variety of models of memory. So, what functional evidence do we have of pattern separation and pattern completion in the hippocampus? Well, there is a really large literature on this that many of you in the audience have contributed to that I don't have time to cover in full today. But some of my favorite evidence in rodents comes from the results of geometric morph experiments. In these experiments, a rodent is familiarized with two differentially shaped environments, here a circle and a square. After this familiarization, they are tested in parametrically deformed versions of these familiar environments. Now, these morph environments can be thought of as, in some sense, linearly stepping through the input space, separating the familiar environments. The classic result looks like this. The maps in the morph environments continue to resemble the most similar familiar environment until some point in the morph sequence where you see an abrupt transition in map similarity, and now the remaining morph environments appear like the other familiar environment. Now, if map similarity alone were driven by the input geometry alone, one would expect linear steps through this space. Deviations from this prediction can thus teach us about the extent of pattern separation and pattern completion taking place. For example, here the two maps are very decorrelated, which corresponds to a high degree of pattern separation. If instead pattern separation were weak, we would expect a higher correlation between the familiar maps. Furthermore, in both of these cases, the transition between the two maps is rather abrupt, indicating a high degree of pattern completion. If pattern completion dynamics were weaker, we would expect a more gradual transition, such as this. And finally, you could always have a combination of these phenomena. Now, what I'm showing you in the initial example is a schematic of the classic result. But follow-up work has shown that these dynamics are sensitive to the particular training experience of the navigator on a short timescale, suggesting that this task might be ideal for mapping the development of the hippocampal representations over protracted experience. So we sought a version of this paradigm that could monitor the hippocampal representational landscape during a steady stream of experience. And due to limitations in calcium recording, we were only able to thoroughly record one environment per day. So we settled on the following paradigm. First, mice were familiarized with two rectilinear environments, a square one and this funky one we call the glen. And you could ask me why we went with these shapes later. After this familiarization, mice were first recorded in each of the two familiar environments over the course of 2 days, while receiving additional unrecorded top-up experience in the other. Next, the mice were recorded in each of 4 morph environments in a random order over the next 4 days, while again receiving additional top-up experience in the familiar environments. Afterwards, the familiar environments are repeated again to bookend this morph sequence. We could do this over and over again, aiming for 32 days of testing, at which point we have 5 full bookended morph sequences. So what do we see? Well, within sequence at the single cell level, we observe a good mix of effects mimicking some things that have been observed before. For example, here are 7 simultaneously recorded example cells to give you a feel for what's going on. There are some cells which exhibit abrupt transition in the preferred locations in the middle of the morph sequence. There are other cells that exhibit a punctate place field in only one context, but not the other. Still others continue to fire at geometrically similar locations across all environments. But these are, and while these results are exciting, these are only individual cells, of which we have literally hundreds, so I could show you a lot of patterns. What is the population doing? So here's the first morph sequence for mouse 1. It looks kind of similar to the classic result in that there's an abrupt transition in map similarity near the midpoint of the morph sequence. But the blue and the red lines are not very far apart, suggesting that these two familiar environments are not maximally separated. With the second sequence comes more separation, and more with the third, and so forth. And in a second mouse, we could see a similar story of progressive separation over sequences. However, in this mouse, we also note that the point at which the abrupt transition occurs shifts over the course of these sequences to be more and more near the glen over time. Now, of course, we could quantify these effects if we want to, and I'm doing so here just because I can. So here you could see progressive decorrelation of the maps with time, and here you could see us mapping the transition point over these sequences. And while these results are pretty exciting, the data are far richer than this alone. So far, I've only shown you a subset of map comparisons, comparing morph environments to the familiar environments only. Instead, here I'm showing you the pairwise similarity of all hippocampal maps for all the recordings for 3 example mice. Clearly, there is some structure in these patterns, and we can pull it out with dimensionality reduction. So this is what happens when you reduce data down for this one example to 2 dimensions with classic multidimensional scaling. Now, these plots are complicated, so let's unpack it a little bit. The color indicates the environment. from square-like in red to Glen-like in blue. The size of the dot indicates the date of each session, with the first sessions being depicted by the smallest dots and the later sessions being depicted by the largest dots. So here I'm circling the early sessions. You could see that they're somewhat clumped together by color, but tend to be closer together, while by later sessions circled here, the separation between these two clumps is more pronounced, mimicking the effects we saw before. Furthermore, you'll note that the representational changes over time all seem to point in the same direction, indicating a continued representational drift. This is true in all of our animals, and here I'm just showing you 3. Notably, the drift axis is orthogonal to the axis on which context appears to be represented. And I think this is a pretty incredible observation, so I want to linger on it. I think the ways that the brain copes with, or more likely utilizes, representational drift is a key question confronting our field, especially now with the advent of calcium imaging, which has made it clear that these representations are a lot more drifty than perhaps we had expected before. But in experiments where only one environment is recorded, it's impossible to separate what might be a drifty temporal representation from the representation of spatial context. To do so requires mapping the representational landscape over time and to some degree of precision by assaying multiple environments, like we've done here. This result indicates that drift does not independently perturb different sections of this representational landscape. Rather, contextual representations drift in concert with one another. Because of this, it's easy to cook up a downstream reader that is sensitive to context, but not drift, and vice versa. Super dope, I think. I think this result is nuts. This observation also suggests that we can decompose these population level data into distinct components, drift and context. So this sort of decomposition is especially useful because it allows us to test whether the activity of individual cells, which we can typically only record on a subset of sessions, if it best resembles one or a combination of both predicted matrices. Now here I'm showing you the matrices of 2 example cells, where the white rows and columns indicate sessions on which this cell was not successfully detected. For each cell with enough comparisons, we can try to explain the cell's similarity matrix on the basis of these predicted drift and context matrices. When we do this, we see that cells tend to load more strongly onto one of the 2 components, but not both, with many cells exclusively correlated with one or the other. So let me explain what I'm plotting here. Again, here, each dot is a cell. The size of the dot indicates the amount of variance explained when both the drift and the context factors are included in a GLM of that cell's similarity matrix. The position and color indicate how much each factor, context or drift, contributes to that explained variance. Now the star is the same analysis repeated on the median population response. So noting a few things here. First, note that cells tend to be explained purely by the context or by the drift, leading to the bimodality we observe here. Also note that individual cells do not resemble the population average, which is a mixed response. Importantly, the characterization of the population as a whole indicates that this bimodality didn't have to be the case. The cells absolutely could have resembled partial loadings on both factors. These results are consistent across mice. And I think it's pretty wild to see this, and it was honestly unexpected for me, because these results are suggesting that individual neurons are contributing to either a representation of spatial context or a representation of time. And this sort of effect can be seen especially clearly in some example cells. So here I'm going to show you a cell that loads pretty purely onto the drift component. I'm showing you here the first morph sequence, and I'm ordering these trials by the order in which they occurred. So you could see this cell kind of fades into existence towards the end of the first morph sequence. In the second sequence, its field kind of settles in the top left regardless of the environmental geometry. And then it kind of fades out in later sequences, a pretty pure representation of drift. On the other hand, here's a simultaneously recorded cell which codes for context with little effect of drift over time. Here I'm showing you the first sequence, and now I have ordered the each recording according to its geometry for the ease of interpretation. You could see that this cell tends to have a field in the lower right section for square-like environments, but the lower left portion of the environment in the Glen-like environments. Now this is the first morph sequence. We see something very similar in the second sequence, and indeed for all of the sequences. I mean, I think this is pretty beautiful. This might be my favorite cell that I've ever recorded. It's 31 of 32 days of testing tracking a hippocampal representation. Pretty cool. Anyway, so this is, in the interest of time, I'm going to wrap it up here today. So to wrap it up, I've told you about 3 projects of mine addressing how experience yields rich dynamics in hippocampal and entorhinal representations. In part 1, I showed you that the grid cell representation is not static in deformed environments, but rather shifts dynamically with the experience of the navigator. And human spatial memory follows a similar pattern. In part 2, I showed you that the hippocampus represents recent experience, even in the absence of sensory cues and explicit task demands. Moreover, this representation is specifically mediated by trisynaptic circuitry. And in part 3, I showed you that hippocampal pattern separation and pattern completion are not stable, but develop richly with experience. Furthermore, contextual representations and representations of drift appear to comprise distinct components mediated by distinct neural ensembles. Now, this work sets the stage for future causal tests of the circuits which might mediate each of these components. And that's what I got. Thank you so much for your time and attention. And again, thank you to the organizers for having me. I'm super happy to be here, and I'm really looking forward to your questions and feedback.
Thanks so much for your talk, Alexandra. Those results were beautiful. The Q&A section filled up quite a bit, so I'm going to ask questions in a chronological manner. So the first question was asked by Amelia in regards to parts 1 and 2 of your talk when the animals were in the same environment across your experiment. So her question is, can you discuss the stability of these measures? For example, how quickly do they emerge and how stable are they over time? Amelia says that they were trying to understand how habituation and even learning can influence these measures.
Okay, is this specifically for part 1 or is this for part 2?
They asked it during part 2, but then added a comment that she's asking in regards to parts 1 and 2.
Okay. So this is a great question for both parts. So I'll start with part 1, the grid cell results. In the data sets that we had, unfortunately, we didn't have a lot of across day data. We had some amount of the across day data from the Barry data set. And I looked at if I could see a day-wise component in the amount of shifting we're seeing of the grid cell representation, and I didn't see an across-day effect, which you might be thinking of this because they showed that rescaling was diminished across time, but I didn't see any sort of effect mimicking that in the amount of shifting. And I don't know what to expect, whether I would expect shifting to be deteriorated or not in this case, but I didn't see it there. Now, in the case of the second part, the data set for the entryway remapping, we recorded most of these mice across 6 days of experience, and they were completely naive at the beginning. And 6 days isn't a lot of time, but they did continue to have this representation from the first session to the last session. And in some supplemental data that I didn't present here in a larger environment, we recorded some mice out to 21 days. And for those mice, the entryway remapping qualitatively looked like the effect size was slightly diminished over time, but it still remained well detectable even after 21 days of recording. And all that data is in the supplement. So if you want to check out individual animals or across time data, it's all there. And the data are publicly shared, so you could also check it out yourself.
Great, thank you. Okay, so the next question comes from Andre Bukers in regards to part 2 of your talk. They're wondering if like really what is the usefulness of the differentiation that you see dependent on the entryway. Someone else responded and suggested that it maybe is just latent learning, sort of like a just in case type of learning that may fade away with experience. So do you have any thoughts on that?
Yeah, so this is a great question, like why bother representing these things? So I guess there's like a top down and a bottom up explanation for this. From the top down, the explanation we kind of went with was the latent learning. You might want to represent this stuff because in the environment, you're not sure what sort of contingencies are going to happen in the world. Maybe someday you realize that if you leave through one door, you're more likely to get attacked by a predator. And if you leave through another one, it happens to be safe. And this is the sort of information you'd want to be able to represent quickly. So it's possible that that is the case. You might make a reinforcement learning, like build a reinforcement learning model explanation surrounding this sort of thing, where you're trying to explain transitions of states. And so in this case, you might want to represent state transitions originating from one door versus the other door separately. In terms of a bottom-up explanation, we noted that in linear track environments, you wind up with more dissociated representation at each of the doorways. So by the time the animal gets to each of the doorways, you wind up with very different representations within the hallway. And so we kind of thought, given this different representational starting point, we would expect some amount of perseverance when the animal has entered the room. And so those were some things that were on our mind. But it's a great question of theoretical interest. So I would like to see some more explanations of this.
Great, thank you. Okay, another question about part 2. So Sang-Hoon Kim is asking, in regards to the Cree line of animals, are you inhibiting both DG granular cells and CA3 pyramidal cells, or only the DG granular cells? And as a follow-up, if you're only inhibiting the granular cells, have you checked whether inhibiting DG-CA3 circuit could also affect remapping of the CA3 pyramidal cells?
Yeah, so story of this mouse line, we started using it because we wanted to specifically target CA3, but the problem was non-specific Cree expression in the dentate as well. And so we wound up with some dentate expression, but ultimately we are inhibiting both. And you could check out the supplement for some more quantifications of the cell counts in each of these regions, but it's not specific to just CA3 or the dentate like we might want it. And I think future work, it would be great to investigate something like that. Yeah.
Great, thanks. Okay, so I think those are all the questions from parts 1 and 2. So now to move on to part 3, Jeff Stott is wondering, how do you think pattern separation and pattern completion relate to your drift parameter?
Great question. I don't actually know because I haven't quantified it yet. I will say that, how do I want to say this? I think I don't have a ton of mice, and so I'm trying to think of within like individual mouse variability. And there are some mice, like my best mouse is also one of the least drifty mice. And by best mouse, I mean the one showing the biggest pattern separation. And so that hints that there might be such a relationship. But right now I only have like 4 or 5 mice, which is good enough for a lot of these analyses. But to look at cross mouse effects, I wouldn't put my money on it yet. So I think it's something that I will be investigating as I continue to record some additional mice here. But it's provocative to think that there is indeed a relationship between them. And one really cool thing that I have considered is trying to do these sorts of quantification of individual mouse differences in pattern separation, pattern completion, and drift, and then test these mice in a different behavioral paradigm afterwards to see if we do see correlations of these behaviors or of these representational characteristics with the behavior of the mouse later on. But I don't have data to show you about that today.
God, it sounds like there's room for future work on this.
Indeed. I mean, I hope there's ROM work, so we'll see.
Yeah. Great. So our next question is coming from Steve Siegelbaum. In part 3, Steve is wondering whether you might provide behavioral relevance of the place cell properties using a fear conditioning paradigm in one context and probing freezing in morphed contexts.
Indeed. So this is a great question, and it's something that I'm pursuing in a separate cohort of mice now, not specifically fear conditioning. And the reason why I've been avoiding fear conditioning is just because of concerns about the sampling of the animals that I would get while recording. But I'm doing this in an appetitive task now, where we pair reward locations with each of the two environments, and then try to assay the reward choices or the choices that the animals make in these morph environments. And supposedly we should be able to track these things. It's also a great question to ask, would the addition of a behavioral task that depends on either separating or uniting these environments, would that lead to increased pattern separation or pattern completion? So this is another idea floating around with how we could use this paradigm. But it's a great question.
Great, thank you so much. That's the end of the questions that were posted in the Q&A. If any of the participants have lingering questions, we do still have a few minutes now. So if you want to pop them into the Q&A chat box. Otherwise, at 1 o'clock, Alexandra will be moving over to the trainee Q&A session. The Zoom link for that has been posted in the Zoom chat. So again, thank you so much for sharing all of your results.
No problem. Do I go to that Zoom